How do you find that? Testing, 1, 2. You have to turn it on. Okay, I'm in the process of, as I mentioned before, removing the blue background to the slides, and as I do, I look at them and see whether I can make sense of the text that's on there. So I'm making little adjustments as I go along. In some cases, I'm adding figures. I'm going to molecular biology of the gene and scanning a figure and then redrawing it and adding stuff. So they're still in revision, and I will put there on the website when they were updated, so you can look and see whether yours has been updated or not. There won't be major changes. If there are major things, I will say be sure to update the latest PowerPoint before the exam. Much of it is cosmetic, and hopefully it'll be so you can print it out without using up all your blue ink. Okay, a new topic. Evolution could not happen without genetic recombination. When mutations occurred, it would not be possible to separate favorable from unfavorable changes. Ultimately, the chromosome would accumulate so many deleterious mutations that it would fail to function. By shuffling genes, recombination allows favorable and unfavorable mutations to be separated and tested as individual units in new assortments, provides a means of escape and spreading for favorable alleles and a means to eliminate an unfavorable allele without bringing down all the other genes with which it is associated. This is the basis of natural selection. So where did recombination, that seems like a rather distant, far off objective of evolution, but evolution acts locally in the present, right? It's selecting for right now. And so how did recombination become such a even necessary part of higher organisms? It's even a part of bacteria, but they can certainly get along without it. How did it arise? And I've been thinking about this a little bit, and as I probably mentioned before, I think it came from DNA repair. DNA repair is an immediate need. Survival of the organism, the ability to pass on its genes is closely linked with the ability to handle a mutation, a thymidine dimer. We have all these complicated mechanisms about a polymerase. One has to stop. You get retrieval repair, where it takes a piece of DNA from the new strand it just made and replaces it. So I think those kinds of complicated repair mechanisms provided the gristmill for recombination, learning about recombination and how to do it as prokaryotes. Of course, now with us, you have to have a recombination before you can make a gamete. It's become linked in as absolutely essential part of going forward in the generations. Types of recombination, we'll start simple here. Homologous, which means two sequences that are the same, they can recombine. We've talked about that with bacterial conjugation, how it just happens. E. coli can't help itself. It also happens when during the resolution of the genome. You know, if you replicate the two copies, you'll get sometimes right at the end. A spontaneous homologous recombination will occur, and you have to resolve that using the XER system at the diff site. So recombination in that sense is a way to repair, to get out of a bad consequence of replication. So homologous recombination, in eukaryotes it occurs at meiosis. In males, this is the meiosis, of course, is the formation of the gametes, reduction division. Mitosis is just somatic cell division. It happens in meiosis in males and in females in the formation of gametes. The other big category is site-specific. It can only do a recombination event when there's a certain sequence there, because it has a very specialized machinery which recognizes it and then performs a recombination operation. That's what happens in the ZIR-diff system at the end of replication if it needs to. It occurs only at that diff site. So some other examples of site-specific would be transposons when they jump, transposition. Specific transposases recognize specific sites to cut. The immune system in us, it goes all the way back to fish, to vertebrates. Vertebrates have this antibody shuffling capacity, where you're actually rearranging the sequences in the genome, in the somatic cell, not the germline, but the somatic immune cells. In yeast, there are 3 kinds of yeast. They have not male and female, but they have alpha and A. And these are both haploid. And then when they join together to form what we would call a zygote, that would be the 3rd form of existence. But that region of the genome, actually you rearrange the sequences of DNA through a recombination event. So homologous, general, which is another name for homologous, depends on complementary sequences, the same sequences. You have to have at least 50 or so before it will work, but it'll work any place, and then site-specific. What kind of recombination is this? This, of course, is phage lambda, and it goes in at a particular case, a particular site, so that's a phage, in this case, would have a site-specific. More examples? That GoPro, he thinks it is lecturing. GoPro, take a hike. It loves to hike and swim and fly. More examples of site-specific recombination. VJ joining, this is what happens in the immune system, in the B and T cells. And you get a shuffling of genes at several different levels. As you see here, it's probably a little hard to see, but this represents the DNA, and you have these variable regions and constant regions of the same protein eventually as you get down here. But you have these intervening areas, exons, and some of them are spliced out through recombination events. There's a little sequence that well, it looks like it's contributing to its pension plan. It says it's saving. What's to save, man? I just love these things. This one's waterproof and it's about to be tested. Okay, so you have this shuffling and it's site-specific. And then at the second step, after you make the RNA, you get a shuffling at the splicing. You get various alternative splicing outcomes. So you get a lot of variability there. This is the yeast mating type switch. Again, example of site-specific recombination. This is where they go from the alpha to A genotype. Homologous recombination can occur anywhere, of course. You just need to have homology among the sequences. It does not restrict it to any particular region. There's no change in genome organization generally, but you can get, that's kind of a misleading statement, but you don't get inversions happening and big shuffling events occurring. It's just usually a splicing at a homologous region. Again, generalized recombination is homologous recombination. We're going to talk about what happens in eukaryotes first. This would be recombination PowerPoint 1, and PowerPoint 2 will be more into the details of the machinery in bacteria, where we learned about how this stuff works in more detail. So if you see 2 sister chromosomes and you see the chromatids there, there'd be a total of 4. A crossover only involves 2 of the 4 at a time, So the maximum you can ever get crossing over from these 4 would be 50%. Only 2 can cross over, and here you see when they cross over, blue becomes red. And so if you start out with the dominant and the recessive, you would get the recombinant would be start off with large A, could end up with little b, lowercase b, and vice versa. No recombination, they would remain either capital or small letter. Whether it's capital or lowercase doesn't mean anything in this context, just a way to separate the 2 chromosomes. Bivalent contains 4 chromatids, 2 from each parent. Meiosis only 2 of the 4 undergo recombination. A single recombination event can only produce 50% recombinants. This is an important diagram. This shows the stages of gamete formation of meiosis, and you see leptotene, zygotene, pachytene, diplopene, and then the last one is diakinesis. It's not one of the teens. What you need to know is in which one of these stages do you actually initiate recombination? And it's earlier than people originally thought. The early microscopists looked through the microscope and they could see what appeared to be chromatids forming these sort of X-like structures. Of course, the Greek, it looks like the letter chi, and so they would call them chiasmata, these X's for chi, crossover. And they always thought that this is probably where crossover is happening, but you wouldn't see that occurring until way down in this region here, actually in this region here, diplotene. But now we know that the double strand break, all recombination starts with a double strand break. That seems kind of dangerous, doesn't it? Double strand break, you got an end, you could go any place, but you have to cut both strands on one of the 2 double-stranded molecules. The one that's going to donate the strand, of course, has to be the one that's cut. So you have 2 strands, one of those duplexes has to have a double strand cut in eukaryotes and in bacteria. That event occurs way back up here in leptotene. And so the original diagram that came from an early edition, I think your current edition doesn't even have this panel here. We had to move the double strand break from zygotene all the way up to leptotene. These chromatids are very far apart. And so what's happening is at the invisible level, at the micro level, there are actually strands of protein, pre-synaptonomal complexes beginning to form, and it's lining itself up and getting ready for recombination at the very first stage, leptotene. Notice what's happening here. It appears to be a zippering up, a pairing, and here it's complete in pachytene. Zygotene, the zippering begins. Then you get a pulling apart, but they remain attached to form these so-called X or chi structures in diplotene. And finally, they're resolved. But this process here, the pairing, is what the early geneticists thought was where the action was. You pair them, so now they are homologous and they can exchange genetic information. What we now know is it's happening already up here. In fact, the purpose of this complex is called the synaptonemal complex. The purpose of it is actually to stop further recombination because it locks them in a configuration where they can no longer exchange genetic information. It's just opposite from what you would think intuitively. Recombination's complete by pachytene. Even though you don't really see the chiasma forming, chiasma, here's the word, until the stage after that. diplotin, and that's, you see it when they begin to pull apart. Human females oocyte meiosis stops at birth, it pauses, and then begins again at puberty. In males, it's going 24 hours a day, 7 days a week. Okay, so more detail of the events here. Leptin chromosomes first visible, DNA replication just finished. And already you get the double strand break to start to trigger recombination. Zygotine, you get a pairing forming a synaptonemo complex, which you see it spelled out here. The zippering up, forming the synaptonemo complex, is complete by pachytene. Then you begin to separate and you get stuck at these regions which seem to correspond with areas of recombination. Double strand break repair. What we're going to do next is go through a series of models that describe the different types of recombination events at the molecular level, at the detail of looking where it's following each strand as it participates in this event. And you'll probably be surprised to find that it's a lot more complicated than you think it ever should be. You're going to need to be able to differentiate between these different models on an exam, so that means it must be the most important thing on earth. So here we go. This is the most complicated double strand break repair model of homologous recombination. Break repair, there's that word, repair. Repair probably is how all recombination started evolutionarily. It starts with a double-stranded break. And this happens both in bacteria and in us. The details of the players are a little bit different. So, first thing, you have two duplex DNAs lined up. Now, they used to think they were lined up because they had the same sequences, right? Homologous recombination. But in eukaryotes, everything is wrapped up in histones, right? If you had to unwind the chromosomes and say, oh yeah, this looks like the insulin gene, what we got over here? Nope, ain't got to it yet. Nope, oh, wrong chromosome. How are you going to pair things up without unpackaging everything? It turns out in eukaryotes, we skip that stage. We kind of do condensed version of it. We decorate the condensed chromosome with proteins in a particular pattern in the heterochromatin that forms, in a particular pattern that is unique to each of the chromosomal arms. And so it's really a protein-protein pairing that has to happen, which is much simpler than having to read the whole genome to figure out where you should be. The underlying genes determine the patterning of recognition proteins that decorate the condensed chromosome. More about that in just a minute. So let's take back up here a little story. You get double strand cut, and then the next thing to happen, and this is a key term, or it should be, resection. I've never heard that word except on slides in recombination. It means specifically where you chew back the 5' end and expose that quivering 3' end. 3' end. That is really, really exciting to what? DNA polymerase. It goes 3' in. Are you kidding me? I can't make my own primers, and there's one just waving in the wind. I want to grow. I mean, like I can add nucleotides and stuff on that thing. 3' ends are really, really special. They are baby gussied up and presented 3' ends. They are red hot. You form one of these 3' ends, you've got yourself a primer. DNA polymerase gets really, really excited by seeing that. That primer, right here, this one, that 3' end is formed by the process of resection. One of these now invades the second duplex DNA, and that is by sequence homology. How does it do that? DNA can't read DNA, but unless they're opened up single-stranded, right? But that's actually based on base content, and you have to have at least 50 bases the same between this protruding 3' end and the recipient strand, and it's done with a chaperone protein. The chaperone protein makes it all possible. It stretches out the single-strand form of DNA and lays it on sort of a scaffold that makes it kind of stiff so that you can now compare with the target. Rec-A actually, 6 Rec-A subunits are needed to go around DNA. It sort of spirals around the DNA. Takes 6 to go all the way around, and the DNA kind of lays in the groove that's formed. It's got sort of a scaffold to present the single strand so that it can be matched up. Rec-A is involved right here, and look what you get, our old friend, the D loop. It's displaced by that event. It's easy to say and to draw, but just think at the molecular level how intricate and marvelous that is probably going to be when we know more about it. So this is the original 3' end. It's a great primer, and sure enough, it's used as a primer. You get DNA synthesis. See this connection between DNA synthesis, replication, recombination. Recombination. DNA synthesis is a vital part of it. All the R words, what? Replication, recombination, repair. So you get what's happening. This blue molecule is now being extended, but it's not using blue as template. It's using red as template. So if there's a single base mutation between these two alleles, it is going to incorporate it, and it's going to be attached to the original one that was blue. The next thing to happen, second end capture. So you've got the other one here. Let's see, nope. The 5' end is called end capture. This comes down right here, and they are ligated. But notice what happens to the remaining 3' end. Instead of protruding and seeking out the other duplex, it is still used as a primer, but it just elongates on the displacement loop. Again, the same effect. It is using its template red. Here you see, so if there's an allele change here, it's going to be incorporated now into what was formerly blue. So both of these 3' ends that are generated here are used as primers. One of them undergoes strand invasion, and the other one is elongated in place. Secondly, a 5' end actually sort of invades or gets attached to the red duplex here. So what you'll end up with are 2 of these little X's here, recombinant joints. So at the end, well, I missed a step here, see the little arrows? This right here now can move back and forth. It's kind of like a slider. You could take that, put a napkin ring around that, and you could just slide it back and forth, and the further you slid it this way, the more you would lay down blue in the place of red and vice versa. You started sliding it this way, you get a whole large region of red that's going to be moved up here in hybrid with blue. So you start with a little event over a short area, and then by branch migration, you sort of like a zipper motion, you move it to the right here, and red continuously replaces the blue, and down here, the blue is substituting for the red. So you can get a very long region eventually due to branch migration, and that can go for 1,000 or more base pairs. You can really spread that region of duplex. And then when that's over, there's the last step called resolution. Resolution has two possible outcomes. One is they can separate, so blue is still connected to blue. No crossover. Yet this middle part is duplex, is hybrid. But it's just a little patch, right? This is called patch recombination. But blue is still attached to blue. No crossover. The other outcome is possible when you cut this, is that blue ends up being spliced onto red, just based on how you cut that. This is called crossover recombination. Both are possible. Patch, which is no crossover, and splice, which is a crossover event. This is the most complicated form and the most conventional kind of recombination, where you have these two outcomes. At the end, you have double-strand break, chewing back of the 5' end, resection, excuse me. You have strand invasion of one of the 3' exposed ends. You have second end capture. That's a specific phrase. That is not in all the other pathways. It's a capture of a 5' end. It's not used as a primer, but it's just attached through ligation to what was previously extended here. Second end capture, and then you have resolution. Two possible outcomes. Double strand break repair. The rest can be understood in terms of this, whether they complete it or not, and how they actually differ from it. Double strand break in one of the DNAs, 5' in resection. Single strand invasion of one of those 3' ends. Synthesis on both strands, down here and up there. And then finally, end capture, and then finally, ligation and resolution. Two outcomes. Okay. Double strand break repair. Again, this talks about some of the features in more detail here. A D loop is formed, displacement loop, as a result of that, of one of the strands invading. This structure is called a recombinant joint. It's not the same as a holiday structure. By twisting it around, it can be folded into a holiday structure. But that per se is not a holiday structure. Five' end capture. That 5' end that is here bends down and is ligated. Recombinant joint, one strand DNA crosses the other, and then this can be rearranged into a holiday structure, and this is what the holiday structure looks like. This takes some folding and flipping around and inverting to make this happen. It's like origami. It takes a little mental gymnastics, but you can rotate the bottom and flip, and it's like a Rubik's Cube. You can form that recombinant joint structure into a holiday structure here. That holiday structure is very precisely determined because it lays on the surface of a scaffold of protein that is configured just to form this, just to make this happen. And why does it want to spread it out in this nice, organized way? Because this prepares for resolution for the cutting event. Because sometimes in meiosis, there's a stage where you want it not to have crossover, and then the last stage you want crossover to actually happen. And how you control that is by which of these places you cut. If you cut here, you have one outcome. If you cut there, you have a second outcome. So, see this molecule here? The white goes down here. If you still have this being formed, although each of these looks like a different one, but let's say this is the blue one. If you cut this way, blue remains attached to blue. If you cut up and down, blue becomes attached to red. The coloration here is not convenient for explaining this because they made every single arm there a different color pattern. Branch migration. Of course, this kind of simplified diagram. This is, I often have an exam question, and they're probably homework questions based on this, and I ask you, do you get crossover or not? Or I'll have arrows going like this and like that, and I'll say configuration A or configuration B, or blue arrows or red arrows. Which of the cutting configurations will result in crossover? So, and I'll have things labeled. So if this is A, obviously if you cut here and here, A is going to remain attached to A. No crossover. Remember, there is that little region though through branch migration where you've actually switched the strands. That is a little patch. That is a duplex, a chimeric composed of strands from each molecule. But it's just a little short region. In the end, you still have DNA A still attached to DNA A, so it's considered no crossover. Cuts on the same strand. It leads to patch recombination, no crossover. If you cut the other way, where A now is joined to B, then cuts on different strands, you end up with crossover recombination. I'm not going to ask you the words, because I think the words are a little, same strand, different strand, you go, which strand? Just be able to visually look at it and tell me whether you're going to get crossover or not when you cut one way or the other way. I'd rather you have, I think, a deeper understanding of it if you just look at it and watch it happen. Okay, so this is some of the machinery involved in a eukaryote. There's a topoisomerase. You know, topoisomerases are able to take and unwind the tension or put tension in DNA. Topos usually put it in. But it has to make a break on one strand and move the second strand through the gap and then seal it back again. It's a pretty complicated enzymatic mechanism. One strand crosses over the other. The top strand actually has to be cut and the strand underneath has to be brought up, and that's how you get rid of one of the windings, one helical turn. That mechanism has evolved in one of the topoisomerases, so it's no longer a topoisomerase. It's used to make the double strand cut. You remember a lot of these things when they cut, as part of their mechanism, they become covalently attached to the 5' end. Here's another example of it. The eukaryotic protein enzyme that does the double strand break is SPO11. Keyword. It remains attached to the 5' end on each of these. That triggers the next step, which of course is resection, so that you have the 3' end at both sides of the cut. You get this protruding 3' terminus, which is perfect for DNA polymerase to use as a primer. So what do we have here? Bob's combination-like sequence, only known mechanism for nucleic acids recognize each other, yada, yada. Note recombinants have hybrid DNA. Okay, so whether you get crossover or patch, in both cases, there's at least that little region in there that's going to be a duplex comprised of one strand from one molecule and the other strand from the second duplex. Whether it actually crosses over their full length or not is dependent on where it cuts. Allelic conversion. You're going, oh no, my head is hurting. I hope I would never have to learn words like this. Allelic conversion. I don't want to have cute parlor conversation. I don't need to know this. Let's try to get through this and be happy on the other side. Allelic conversion, that's where you have two alleles. Let's say it's an enzyme, and one of the alleles has lost enzymatic activity because there's a single amino acid mutation in the active site. They're essentially the same gene, except, it could be one nucleotide. And would it be a synonymous or non-synonymous mutation? Non-synonymous, because the mutation results in a different protein, a different amino acid being inserted. So you've got one nucleotide difference, and of course that's going to be at a specific region on the allele. But let's say it's right here in that region that's, for some unlucky reason, was subject to recombination. It can happen any place, homologous recombination. What you'll end up at the end is, it will appear as if by magic that when you sort out the gametes, it's as if you've converted one allele into the other allele. You've lost one, like one got switched. And that's exactly what happens if the recombination event itself, where you get that hybrid duplex, if that happens to occur in that region that is different between the two alleles. So let's see how this would work. There's a fungus called, I believe it's Neurospora, very famous. It forms an ascospore, and the order of the spores, it has 4 spores. You can follow the fate of each of the 4 strands in the, what's it called, the tetrad. When you have the 2 alleles, and you have the 4 chromatids, you can follow a mutation, and each corresponds to a particular strand. You can just count those, and you can tell whether recombination has happened. The normal distribution is 4-4. That's the parental ratio, and you'll get 4 coming from one parent, 4 coming from the other. If there's no recombination, that's the ratio you're looking for. Anything else raises a suspicion, especially on an exam, that you have allelic conversion. A 3 to 5, where you got 3 and then 5. It's as if one of these blue got turned into a red. Allelic conversion. Here's a 2-6. It's like 2 of the blues got turned into red. They were converted, changed over. And it's because of the repair operations that occur after this duplex region is formed. So If the region where the two alleles is outside of the area that undergoes the actual holiday structure and branch migration, if it's right here, you'd end up with 4-4 again. If there was nothing you could screen for, then you would think there was no allelic conversion, because let's say there's only one nucleotide difference, but it's located here. If you got a switch, a recombination here, and then there's no repair to happen, but since they're both the same, there's no converting. You still get the 4-4. Ascomycetes, tetrads. Each spore represents one DNA strand at meiosis. Unusual crossover ratios. Crossover within the allele, if no recombination within the allele, is going to be 4-4. So on the exam, you're looking for anything other than 4-4. Then you're scanning the list of multiple choice for allelic conversion must have happened. So you didn't even understand it, and you're making 100 off to med school. Who needs to understand anything? Let's open up his skull. What's in there? Two causes. A gap repair, which right here, this is the gap that's left in the strand that remains behind. That's called gap repair. That 3' end that did not invade, that gap has to be repaired or filled in. And if it'll copy from the red allele. And now the blue becomes red because it copied in the region where the allele was, where the change in base composition was. So gap repair causes it. And mismatch repair, another repair operation. This is not due to synthesis, but the strand invasion thing. You got, let's say the red is mutated, is different than blue, and so one of the two strands is going to get repaired. Depending on which one gets repaired, you're either going to gain a red or you're going to lose a red. Gap repair, mismatch repair. And notice all of this is happening within that region that's actually undergoing the strand invasion and branch migration. That region in the patch that's always going to be the result of recombination, whether there's a crossover or not. Not dependent on crossover. It's in that common part of recombination that doesn't require that there's actually a crossover. Sometimes gene conversion occurs during mitotic. So we've talked about double strand break dependent recombination. All those complicated steps, but they're not that complicated when you break them down. Now we're going into the second general mechanism of recombination. And we talked about allelic conversion. So let's look at the second one here. Notice there's some steps missing. Synthesis dependent strand annealing model. I find this kind of hard to remember. Synthesis dependent strand annealing, that what you should key in on strand annealing model. Let's see how this works. It starts again with a double strand break. You get resection, chewing away of the 5' end. You get strand invasion. This bottom one invades, you get a D loop, and you get synthesis occurring. But strand displacement, and you get a repair, but notice the repair really uses its own template again. There's no, what's missing here, it stops here, there's no 5' end capture. It just does the first half of the reaction, right? Everything's going fine except this 5' end remains up there. You only get one of these the recombinant joints. You actually get I'll show up here again, no holiday structure formed. No second end capture and no holiday structure formed, even though there's a recombinant joint. It doesn't proceed into holiday structure. Strand annealing, synthesis and ligation. It's so sad, it just goes halfway. No crossing over ever. No holiday structure to make the decision which place to cut. Occurs in the first phase of meiosis in most eukaryotes. It doesn't go to crossover. We'll see in a slide that's coming later that this is how things start, and then it triggers the second phase, which is where you get the 5' end capture. It seems to separate it into two stages if you're going all the way to double-strand break recombination. The third mechanism, we have double-strand break dependent, now we have single-strand annealing synthesis dependent, have the third one, and this is non-homologous. The pairing is not based on DNA. It's based on protein-protein interaction. Non-homologous end joining can repair double-strand breaks. Notice where it plays a big role. In the immune system, the V, D, and J joining events. It's actually not based on homology, although there's a common sequence motif, but that's a sequence motif for the binding of special proteins, and the connection is made between the proteins. Here the proteins are the Ku brothers. Ku, heterodimers. Ku70 and Ku80. They bind to each of the two ends. Sometimes there's a little deletion or so, it's not real precise, but what holds them together is not because they are the same sequence, but because they are binding sites for Ku and Ku. Ku70 and Ku80. You get a little repair, you fill the gaps, and then everything's good again after you remove one of the V or J or a block of V and J. First, frequently nucleotide insertions or deletions occur in this process before the ligation step. So non-homologous end joining. You see it? This is what you have to worry about too if you have a chromosome break. This is why chromosomes, since they are linear molecules, have special structures on the end called telomeres to prevent this from happening, because this is a maniac. If it sees double-strand break, it's going to try to put them back together, and sometimes with chromosomes, it can sew two different chromosomes together that shouldn't go together because it's just based on protein-protein interaction, not on nucleic acid homology. Will this never end? Here is what? Number 4. Single-strand annealing function. No, this is the same. Single-strand annealing functions at some double-strand breaks. And let's see, what was the other one called? I get them mixed up myself. Is this the same? No, it's synthesis dependent. Non-homologous, this is single-strand annealing. Begins to sound all the same. So you'll have to take a blank sheet of paper and write this out and make sure you have it better separated in your mind than the aging instructor has in his mind, his shriveled mind. Single-strand annealing. Look at this thing. This can only happen in repeated DNA sequences. A lot of repeats in satellite DNAs and things in eukaryotes. More than half the genome is just repetitive leftover retroviruses and transposons and stuff. That area is subject to this kind of homologous recombination, and this is how it goes. You have two of the same repeat, that's why they call it a repeat. You get a double strand break in between the two repeats, and now you get resection, so you get the 5' end chewed away. And now what happens is this repeat is homologous to that one, so they come together that way, and all that intervening material gets deleted in the process. So single strand annealing. Everything's going along, got the double strand break, except, both of them are being broken. And then the annealing is just in the repeat area, and everything that's not a repeat is just waving in the wind. It gets trimmed off, a little repair, and there you go. 2 minutes to go, result in elimination of non-repeat sequences. Between the two repeats, no D loop is involved. There's no strand invasion going on here. No strand invasion. And here's the last one. Chromosome break induced replication, BIR. And this is where you can get an exchange of the ends of chromosomes. So let's see how this works. It involves this repeat. And notice the repeat is found on two non-related chromosomes, non-homologous chromosome. But it's the same repetitive sequence, the same transposon family invaded both of these chromosomes. So you'll get breakage. Sometimes these repeats, if they involve old transposons, sometimes these transposons wake up from their long slumber, and that transposase gets hungry and starts chomping. And when it chomps down, it makes double-stranded breaks, and sometimes it can chew the end off the chromosome, the arm. Oops, lost an arm. This repetitive area now becomes substrate for recombination, and in the recombination event, you get a duplication. So you get an extra copy of this right arm here of this blue chromosome. And just all of this is lost. All of these genes are lost forever. Repair involves annealing to a repeated sequence present on a non-homologous chromosome. Duplication of an arm onto on one that's lost its arm, but it's a non-homologous chromosome. And here you see the details of what's happening here. This is the original one that had lost its arm, poor baby, and that's a 3' end. In this case, strand invasion is happening. Every time there's strand invasion, there will be a D loop produced. You get a recombinant joint forming. You get a synthesis at that 3' end, and this branch migration happens. But there's no 5' enjoining at all. 5' capture. No 5' capture step. No end capture. Yeah. And that's it. That's enough. For me, this is one of the hardest sections because I have to just memorize this stuff. And by the time the exam comes around, I know this stuff. I'm ready now to look at the questions and see whether they have any mistakes in it. But every year, I have to relearn this stuff because it just doesn't stick on the slippery outside surface of my onion brain. There are no grooves there anymore. Double strand break repair. That was the first one, most complete, most steps. Synthesis dependent. No 5' end capture, no crossing over. Non-homologous end joining, that's pretty easy. You got Ku and Ku. Single strand annealing, that involves repeats, and you remove the intervening sequences, no D loop. Break-induced replication, that's where you copy a portion of a non-homologous chromosome. So this kind of condenses it in your mind to separate them instead of just slide after slide and droning on and on and on. You can finally separate them into distinct topics again as you prepare to have them invade your mind. Involved in allelic conversion, just the top two. Okay. So now that's the details of how the ends of the DNA actually invade and cross over, and you get resolution between patch and splice and so forth. Let's look at the formation of this synaptonemal complex, where the early microscopists started to get excited because they thought they were seeing the structure that made recombination possible. Surely you would need to line things up very rigidly so that you could now effect this exchange of genetic material between the two sister chromatids. A basic paradox is that the parental chromosomes never seem to be close enough for recombination to occur. If you look at the structure of this synaptonemal complex, and here, the synaptonemal complex, if you look really closely, you can see this is really two dark axes, two dark bands. Each represents a structure that is separating the DNAs from the chromatids from each other. It turns out this synaptonemal complex is more of a barrier, a wall, keeps them apart so they cannot exchange. That is triggered after the first recombination on the chromosome begins. The synaptonemal complex then zippers up to make sure it's limited to only one per chromosome. So its role, we know now, is to prevent too many from happening. We now have evidence that initial strand exchange occurs at the great distance of 400 nanometers. What's the diameter of DNA? About 10, isn't it? So this is huge, as they say, in a certain capital city. Synaptonemal complex. Sometimes this is on exams. You have to label parts. It's not just noise. This part up here, the dark swirling clouds, this is the DNA. It's all condensed, but it's separated. This is from one chromatid, this is from the other. So you have the DNA, the chromatin, you have a lateral element, sometimes called an axis. We have one here and one down here, two lateral elements, and a central portion. In the central portion, you have proteins called zip proteins. We'll learn about zip1 and zip2. And you have cohesions down here. Cohesions stick it all together. Oh, well, I got it wrong. The diameter of DNA is 10 nanometers, 2 nanometers, not 10 nanometers. They both start with a T. 2 nanometers, not 10 nanometers. The early part of meiosis, homologous chromosomes are paired in the synaptonemal complex. Each chromatid is separated from the other. So here a diagrammatic representation of what we saw. Cohesions are at the bottom of these loops of protein and DNA, and then in the clear center portion you have the zip proteins. Blocks recombination inhibits formation, blocks in recombination inhibit the formation of synaptonemal complex. But blocks in synaptonemal complex formation do not inhibit recombination. So it's not a reciprocal truth. So if you can't do recombination, you're not going to go all the way through to forming a synaptonemal complex. Recombination happens first. And once that is triggered, then you can, a cascade of events is triggered that leads to the zippering up of the synaptonemal complex. So if you have a mutation that is unable to form this synaptonemal complex, it does not stop recombination from happening because that happens first. That makes sense. This complex is responsible for what is called crossover interference. It keeps the distance between crossover events to be well separated. The result is it's a strong limitation to how many times you can cross over within a particular chromosome. It forms after the double strand break. Double strand break happens, remember it's SPO1, and then what is happening down here? These are some more details as we go from synaptonomal complex, then more details, then back again. We're back in the weeds. So there's some proteins involved here you need to know. These should be key terms. You know SPO11. That makes a double strand cut. Now we're going to learn about a couple more. RAD50 blocks formation, mutation blocks formation of the 3' protruding ends. It also prevents formation of synaptonomal complex. So this is the enzyme, RAD50, that chews back the 5' end, right? Does that resection, because if you mutate it, you can't do that anymore. So this is, if you see RAD as part of the gene name, you know it's a yeast gene. And it's named RAD because of radiation. They found it after they UV irradiated the yeast. So it's a radiation-induced mutation in recombination. So if you mutate this, you can't trigger the subsequent formation of the synaptonomal complex. It's part of a 9-member complex. It's just one of the enzymatic activities in a large recombination complex that exists in eukaryotes. In this case, it's called RAD. This would be in yeast. But I think that language is in an exam answer or question or something. RAD proto, a 9-member complex. Complex of 9 proteins grouped into 2 subdomains. You have your exonuclease activity. This is the RAD50. And then we have the last one we need to know here in terms of the eukaryotic model. So you've got SPO11, does the cut. You've got RAD50, does the resection, chews back, making possible the protruding 3' end. And then you have the last one that has 2 names, depending on where you find it. In humans, it's called DCM1. In yeast, it's called RAD51. It's the next of the RAD series. You'll need to know these distinctions here because this is the same. It's the same. It's functionally analogous to REC-A. Of course, this doesn't mean a lot because we haven't talked about REC-A, but REC-A is that molecule in bacteria that lays out the strands so when you make a 3' protruding end, that it now can be used to pair up and find the correct place to match. REC-A, one of the central proteins of recombination. It forms a scaffold that spreads that single protruding, invading strand out so it can start matching up properly. REC-A, REC for recombination. The functional homologue is RAD51 in yeast. RAD means it's from yeast. DCM1 is what the same thing is called in humans. So RAD50, DCM1, RAD51, SPO11. These are the things you need to know. So how does this, what's the timing back now to the progression of meiosis, the stages? When do these recombination events occur? So we sort of introduce a topic, we dig in the weeds, we get some molecular detail, then we step back and look at it again. Here we're trying to sum it up with a chart that shows the actual time it takes for things to happen. And notice you have synaptonomal complexes formed right at the end of leptotene, So synaptonomal complexes form after there has been a double-strand break, right? So the double-strand break must have occurred here. And then once you get one forming, a synaptonomal complex begins to zipper up very close to where that first break occurs. Synaptonomal complexes dissociate at the end of pachytene, beginning of diploatine. But notice there's a stage here where you don't have crossover products. If you interrupt recombination in eukaryotes, in the earlier stages, nothing has crossed over. Like this cut option doesn't exist. It only exists in one way. That's because you don't really have the holiday structures formed at the beginning. You just have the joint molecules. It has to actually be spread out on that big scaffold structure before you can have the choice to go either crossover or no crossover. Let's superimpose the thing here. Strand exchange intermediates form over long distances. So not only do you get a double strand cut, you actually begin strand invasion very early when the molecules are very far apart. How does that happen? We still don't know. Seems not intuitive. Why it's so far apart? They're actually exchanging DNA and we can't see it. That seems to be true. The chiasma, which the early geneticists saw, happens almost after all the fun's over, where you're actually getting recombinant molecules in the later stages here of meiosis. It actually looks like an X. So this first phase, it conforms to the model that we went over, the second one called synthesis-dependent strand annealing. That was that second model. You remember it doesn't have any 5' end capture. No crossover happens. It's like the first half of double-strand break-dependent recombination has happened. And then when you get into diploatine, you get full double-strand break repair recombination. That's kind of the take-home of this, how that really fits into. So time in minutes. So you can see this occurs over a fairly short time, but not always. You know, in the newt, you have 6 months between prophase and some of the other stages. It gets paused. And of course, in human mammals, not human mammals, in mammals, of which humans are a member, I don't know these female things. Anyway, the females, it starts early, gets paused, and then at puberty it begins again. Okay, so here's just a reminder model, the synthesis-dependent strand annealing, just kind of as a review here, what's happening. This is there, and then just to jog the memory, the full double strand. Well, not yet. Now the double-strand dependent model. 2 independent phases. First phase, second phase. Double-strand break repair, phase 2. Kind of summarizes it up. Early stages in human fetal ovaries. What are these dark lines you see here and here? That's the synaptonomal complex. There, you don't see it so much in leptotine. It's just beginning to form because the double-strand break has already happened, and now you're beginning to get formation of synaptonomal complex. Now you see them beginning to pair, fully paired, then you get the chiasma form. The chiasma represented by the arrows. These little. What stage of meiosis are double-strand breaks first induced? Leptotine. Central elements resumed. Oh, that's a node. You don't see that in all organisms, but this is a indication of what recombination might look like. But remember, the synaptonormal complex is there to prevent recombination. So this is a picture, even though it's blurry, sometimes you'll see it again. It's usually on a late Monday night with a proctor looking at you. I don't know. I don't know why that is, but it seems to be a correlation. This is from onion. It's this early stage, these early strand exchange intermediates are beginning to happen very early with the DNA at a great distance. And once you see these early complexes form, then immediately you will start to see that zippering up here? That's the strand exchange. The double strand cut, the protruding 3' end, the 5' resection, all that's happening right here. But notice what's happening right beside it. See this very rigid, where the spacing is absolutely set? That's the synaptonormal complex being triggered because of the double strand break that occurred right here. And this is to prevent further recombination in the neighborhood. In this region, these DNAs are 400 nanometers apart. But in these strand exchange intermediates, you get these Rec-A homologs, functionally like Rec-A. So it would be DCM1 if it was a human. I don't know what it's called in onion. RAD51, not RAD50, RAD51 in yeast. The zips and hop, and I think that's the end of the first part here. ZIP2, Tippecanoe and ZIP2. Role of ZIP2. ZIP2 binds the protein present in the early recombination. So the first thing to happen is ZIP2 binds to double strand breaks. You remember where the ZIP proteins are? In the synaptonormal complex, that region between the two sister chromatids, that's where the ZIP proteins are. On either side you have the cohesions and then the ZIP proteins in the middle. That zippering up is formed first by ZIP2. It has to go, and once ZIP2 is in place and it is recruited at a break, a double strand break, then it recruits ZIP1, which does the zippering. 2 is unable to discriminate between homologous and non-homologous pairing. It just goes to a break. It's not involved in pairing at all. Double strand break, ZIP2 goes there. Wild type ZIP2 protein localized to the axial association requires double strand breaks. Let's look here. The axial associations hold pairs together before synaptonormal complex formation. So ZIP2, ZIP1, what you see here is, this is wild type. And all of these are in exam 2. No, they're not. I would never tell you exam question. Not even today would I. Wild type, and what would you be looking for? What are these? These are mutations in the zippering up. The ZIP1 and ZIP2, they end up with the same appearance. You're unable to form the synaptonormal complex. So how could you recognize that in case you were so unlucky as to be required to? You see both of the axes, but they're not zippered up. You have an association, homologous pairing, but they don't form this tight zipper. Very loose. You would say, wow, that's obviously some kind of zip mutation, 1 or 2. Can't tell at this level. And I would go, that's remarkable. Where'd you learn that? Open up my skull now. Fix some stuff. You don't need to go to medical school. You're already there. Requires double strand breaks, and these are what the mutations would look like. Pairing happens, but there's no actual tight zippering up like you see in wild type. Seem to initiate, but can't actually make it. No synaptonomal complexes. Note that sister chromosomes are not very close together, but they are loosely paired. ZIP mutations are able to pair, but the synaptonomal complex formation's blocked. It can't go any further. ZIP2 and ZIP1 result in no synaptonomal complex. And the last one, step lively, HOP2. These are the proteins that actually decorate the outside of the chromosome so that one chromosome can recognize its homologous pair. This is that pairing, a part of that pairing identity complex that decorates the outside of chromosomes to uniquely mark them as the particular one they are so they can pair appropriately. This is how you pair without having to open up the nucleotides and read the nucleotides. A mutation in HOP2 has no problem forming the synaptonomal complex because that's triggered by a double-strand break, right? But when you look at the chromosomes that are zipped up, it's non-homologous pairs. They don't recognize each other. They pair up with the wrong partner. Chromosome pairing occurs, but not between homologous regions. Mutations can occur in pairing, chromosome pairing, HOP2, or synaptonomal complex without affecting the other process. Crossover control. Each pair of chromosomes only acquires one or two crossovers, but in most cases, at least one event occurs per chromosome. What this says is, in meiosis, formation of gametes in mammals, you have to cross over at least once, but seldom more than once. And this is how it's controlled by the synaptonomal complex. Sometimes you'll cross over twice per chromosome. But most of the time, one is required. If you don't do that, further development of the gamete is blocked. Recombination now has become a part of how we propagate. Specificity of pairing is controlled by HOP2 protein. They show normal amounts of synaptonomal complex. That's pretty much what I just said. Other proteins involve MSH4, mismatch repair, and DCM1 rad. I don't know whether you're going to need to know that one for this exam, but these two homologs of Rec A, DCM1 rad50, certainly. Would recombination occur between each of the chromosomes, or would recombination between each of the chromosomes still occur in a HOP2 mutant? No, because recombination involves more than just pairing. Recombination is sequence specific. If you're paired with the wrong chromosome, you can't get recombination. You can get the double-strand breaks occurring, resection, but there's no homology. Will recombination still occur in a rad50 mutant? What is rad50? That's that resection. That's before rad51, the one that chews back the ends. That is no, because the slide said it was no. Let's see what it says here. Recombination would not occur since the 3' ends would not be processed, so you wouldn't have a protruding end, so you couldn't get recombination from a lot better reason than I just memorized the slide. And this is the end of part 1 of recombination. What time is it? Is it the weekend yet?
It's the quarter after, so.
Quarter after?
You have till 10 till.
Anybody need a break? There's one guy, everybody look at him. Okay, we'll take about a 2-minute break. And we'll start on the second part of recombination. This is pretty information. I haven't had a chance to switch out the first slide where I have a picture of genes 12 on there. Recombination. This is our old friend. Is this a recombinant joint or a holiday structure? Holiday structure, right? And I haven't had time to remove the blue here. Rec mutants have the same phenotype, unable to support generalized recombination. There are 10 to 20 genes involved in the recombination process in E. coli. Fortunately, we're not going to need to know very many of them. Bacteria do not generally exchange large amounts of duplex DNA, but opportunities present themselves. Conjugation. Now, conjugation is an infection, right? When they're infected by a plasmid, they can't help it. They undergo recombination with that inserted DNA if any chromosome from the donor comes along. Radiation damage, repair. Recombination's involved in repair, the retrieval repair we've talked about a little bit. Phage, another infection, not something E. coli wants to have anything to do with. Repair of a lesion, that is something that it does need to do, and so that's a legit, in terms of E. coli, legitimate use of recombination. And any two double-stranded DNAs for recombination, you have to have a 3' end generated. You have to have it, that's the minimum requirement. 3' end, of course, it involves DNA replication. So let's look at this marvelous system here. This is another little molecular machine. This is the RecBCD. Yikes. This little thing is like a Pac-Man. It's like two Pac-Man. It has it's one Pac-Man, but it chews both strands. So we'll stop with stupid analogies and get on to the details here. This is the figure in the book. And I've tried to complicate it by giving you many forms of this, and we'll go into so much detail that you will really thank me at the end of this class, because now you know everything. RecBCD, no A here. Rec A, we'll learn about, it's part of this eventually. But so what's the deal? The overview from 30,000 feet is this thing prepares the 3' protruding end for recombination. This works in bacteria, and that's its function in recombination. Prepares the 3' end so that it can undergo strand exchange. Its other function is it destroys invading DNA. You get DNA that's not E. coli DNA, a phage DNA, for example, this complex will chew it up. In fact, it chews up E. coli DNA until it comes to a special sequence that says stop chewing and get on with the business of preparing the 3' protruding end. Those special sequences are hot spots of recombination called chi. Everything in recombination is named chi for crossover, for chiasma. Chi, the chi site, C-H-I, I think it's chromosomal hotspot integration or something. So if you have a chi site, this nuclease that chews both strands away stops doing that, changes its function, and now tries to generate a protruding 3' end, and it actually coats that end with Rec A. Pretty amazing. So here you see it. It has two helicases. Not all helicases are these hexamers that form a necklace. These are helicases that don't have that physical appearance, actually. And they go with opposite polarity because you're going along both strands simultaneously. The whole complex is moving in one direction, so one has to be going from 3' to 5', and the one on the other strand is going 5' to 3'. One of them is fast, and one of them is slow. Now just imagine what would be the consequence of that. The fast one would keep going along, just melting like crazy every time it's burning an ATP, but the slow one, it's going to be falling behind, and the DNA that's being opened up by the first one, it's just going to flap around in the wind, right? You're going to get a big loop being built up on one side, and it's going to have to go, wait up here, but wait, bro, wait, it's going to try to catch up, but it can never catch up because it's slow. But that's on purpose. It's forming a big loop on one of the strands and not the other. The top one here, which is Rec D, is the fast one. And Rec B is the slow one. Rec B, though, does lots of stuff. It's a helicase, a slow helicase. It can't catch up, so a big loop of DNA starts to swing down here. And it has a nuclease domain on the back end, kind of spread out on a big linker. And this little Pac-Man structure is chewing this strand and that strand. It's chewing both of them. It's cutting more on this one than the top one. Notice this green thing. This green thing is the chi site. Recombination is greatly stimulated on this side of the chi site. It's directional. It turns out the chi site can only be recognized on one of the strands because it gets separated here, and when the strands are separated, now you don't have duplex anymore, obviously, and the domain that actually looks and goes, aha, a chi site, is only looking at one strand. So the chi site has polarity. You have to approach it from the right. It has to be on the correct strand. And so it's actually on the strand that corresponds to the 3' terminus. So let's just look at the big picture, and then we'll look at some of the ones that are more informed. This is from the book, and this is what it used to be until last year when I started reading and complicating things. It's a nuclease and a helicase activities. This C, you have Rec. I got it mixed up because this figure is inverted compared to the ones that follow, but the one on the 3' strand is actually Rec B. And so it looks like that figure might be flipped. Let's don't talk about this book figure because it's not as informative as the others. There's a chi site, double strand break, B and D. Well, the labels are correct here. Chi site triggers the loss of Rec D subunit and nuclease activity. Yep, probably. There's the chi site. It only is active on one strand. Occurs every 5 to 10 kilobases. You have about 80 of these chi sites in the E. coli genome. The probability of it appearing by chance is about 4. So E. coli genome is loaded up with these chi sites. That keeps it from being destroyed by conjugation, because if there is a chi site, this Rec BCD changes from purely nuclease into recombination machine. Occurs every 5 to 10 kb in E. coli. Orientation point only one direction. Old model. This is the beginning of the new here. This is what I got from a review article 2012. And so this is what you can see the helicase here, you have two helicases, B and D. B is the one that has this split domain. B is also a nuclease. What does C do? C recognizes the chi site right here, and it splits the strands. It doesn't open them up, but it makes sure that the two strands go down different channels. So I'm going to go to the next one, which I think is more informative. And this is what I redrew from molecular biology of the gene, and then I added some stuff. So this to me is, and the one that follows, is the most informative. So let's go through it, and you'll see what a marvelous machine it is. You see the chi sequence, the little green box. Notice that the strands are flipped here to make this easy to understand. The chi sequence is only recognized on the strand, on the bottom strand in this configuration. So the 3' end is underneath this complex. So the chi site is read on the strand that has the 3' end with respect to the complex. You see the fast helicase, Rec D up top, the slow helicase, Rec B. B, remember, has these two domains. One is a helicase, and the second one looks like a little Pac-Man. That is the nuclease. It's part of the same protein, it's just buried in the structure of the middle one, which is Rec C. So we got that. Rec C is involved in channeling the two strands so they go in different directions. The unwinding is done by D, fast, B, slow. Notice as it's going along, this Pac-Man is cutting the 3' end all the time, quite frequently. And occasionally it'll come up and trim the 5' end. See, 3', 5', that's the 5'. It'll cut the 5' end also. So both ends are getting destroyed. If there was no chi site, it would chew the entire DNA up. You wouldn't have any genome left. And this is what it does to invading DNA. But notice what happens. You notice you get a buildup of a loop because the Rec B is so slow. It starts bunching up. Rec B has to go this way. Even if it's not doing helicase activity by separating strands, it's just a little motor that moves one base at a time, whether or not it's in duplex. So but it's going so slow, the loop gets larger and larger. And finally, when it goes over the chi site, and the chi site goes over that little recognition spot on C, Rec C, it causes everything to stop. It pauses and goes, uh-oh, I've been chewing E. coli DNA. Changes conformation. The little Pac-Man stops cutting and starts doing something else down here. What you see is it pauses, and now when it proceeds, the slow one is in charge. It's going more slowly, and the top one is now the slower one of the two. And the 5' end here that's been chewing away is underneath Rec B, D at this point. But now as it starts to move again, but at a slower rate, that 5' end comes out. As it moves along, what you see is the little Pac-Man guy is no longer chewing anybody. Pac-Man has been converted to a productive citizen. It is taking Rec A and loading it up on that big looped out 3' end. It's loading it up with Rec A. This is the machine that loads the 3' protruding in with Rec A, so this can now undergo strand invasion. You may have to label the parts. I have hours going to stuff, so get familiar with how this thing works. This Rec A protein, you put down one, And then by cooperativity, the next will come. But the next comes and only sits down on one side of Rec A. This long polymer grows towards the 3' end. So eventually it will grow and displace it from the Rec BCD complex, and the entire end will be coated all the way out to the end, the 3' end will be coated with Rec A. So it starts off, if there's no chi site, just chewing away both strands, forming a big loop on the strand that corresponds to the 3' end. And there's something left out here. What should I put here? Single strand binding protein is going to be binding this. It's just left out for simplicity. It's going to get displaced by Rec A down here. So that's pretty neat, I think. I think this figure is best. The others are a little hard to follow. Okay, this is from a review article. And again, you can sort of see it. See Rec B, it has this little arm where it has Pac-Man joined to it. Rec C is this big white circle. Well, no, it's a blue circle. And it has channels for each strand. It also has the recognition site for the chi site. So I'm thinking that probably if I clean this up, I should take out all of these except this one. Don't you think? You don't care. Let's just get through this. To me, this tells the best story. It's easier to follow. Although that's not too bad, but it's hard to construct a narrative. Not quite as hard. Okay, this is more about the molecular structure. You will not see this on the exam per se, but you might see something with labeling parts. Maybe there's a label the part on this one. Probably since this is the previous one, maybe I'll make one for this, I don't know. These two are most important for exam purposes. This is just shows you what it actually looks like. Rec B nuclease domain. That's the Rec B was the nuclease domain. Right in here, I guess. No, Rec C, where's the nuclease domain? Okay. So that was the update from last year, plus a little bit of update for this year. Let's see what, for 150 years I taught to make sure all the information is covered. I'm not sure whether this is still in the book or not, but you see the first step is the Rec BCD hands off from preparation of the protruding end, it hands it off to Rec A to facilitate formation of a triple-stranded complex. So we're transitioning from Rec BCD into Rec A, strand invasion. Well, where did that come from? There it is. Rec A. One of the DNA molecules must have a single strand, one must have a free 5' end. These are the requirements, the minimal requirements for recombination that people have worked out in vitro. One of the molecules must be single-stranded. One of them has to have a protruding end. So you can't just have two circular plasmids. Somebody has to have a 3' protruding end to act as primer. The single-stranded region, furthermore, must both be located in a region of common homology. And here's some of the in vitro reactions you can get to go. You can put a single strand, so you need a single strand in, and the single strand end has to be in a region of common homology. Both of these groups of molecules are homologous, and you just put them together. Rec A will bind all around this, and it will replace that and end up with this dark blue strand that was originally on the plasmid will end up free. It'll also do it this way, where the single strand DNA is circular, as long as there's a 3' end someplace. This just illustrates the different possibilities in vitro with Rec A and DNA. Here you see an EM of it actually happening. The single-stranded region is right here, and that's the actual region where it's strand invading. Here's strand invading. Notice the strand invasion itself did not involve the 3' end. There's no end there. In vitro, people try to take this apart. In this case, there is only 274 bases of shared homology. That's why you see it stopping in this experimental setup. How is Rec A able to find the region of homology between these two molecules? How is it able to form the pre-synapsis complex? So these are the stages that people have, I guess, categorized. There's the pre-synapsis. Rec A binds single-stranded DNA. Pre-synapsis, you're getting ready. Rec A binds to the single-stranded DNA. Here you see it. Rec A has bound to that single-stranded DNA. Single-strand binding protein displaces the duplexes and then is outcompeted eventually by Rec A. So you need a combination of single-strand protein and Rec A. The single-strand protein is able to melt the duplex regions so that then Rec A can fully bind to DNA. Next stage is synapsis, so pre-synapsis, then synapsis, and as you might expect, that's where you get the actual intertwining here. They act together, achieve strand invasion, and you have a paranemic and plectonemic joints. They've been shown to form in vitro. A paranemic joint is where they're just pals. They just lie side by side. And this is twisted sister right here, plectonemic. They're really intertwined. Para, plecto. Para and plecto. Know the distinction. What we got? An hour and a half? Okay. It'll feel like it. Paranemic, plectonemic. I think in Greek it means twisted or something. Plecto, intertwined. Heteroduplex extension, once it goes from paranemic then to plectonemic, and then the end is postsynapsis. Stages. I always ask you which is the fast stage. Presynapsis is slow. Synapsis is the fast one. Once you get it aligned, it just zippers up. You get formation of the plectonemic complex. And then postsynapsis strand exchange is slow. This is where you have to actually say goodbye to the old strand and embrace the new. But this part, the synapsis is fast, slow, fast, slow. Rec A. Notice this is more appropriately how it looks. Rec A itself is forming sort of a scaffold, and 6 of these, I don't know if it says 6 here, but 6 of these need to bind in order to make one helical turn of the Rex A molecule. So it's 1, 2, 3, 4, 5, 6. and DNA is laying in the major groove of the Rec-A. Presynapsis. Here you see it up here, where the DNA is laying in the major groove around the outside of that filament of Rec-A. Six Rec-A monomers per turn. Deep groove containing DNA, 3 nucleotides per Rec-A monomer. Okay, these are amazing facts. Rec-A can only bind 3 nucleotides at a time.'Just think how much Rec-A you need. You need, do a KB, a protruding, 3' strand. You need lots and lots of Rec-A. It can only bind to 3 nucleotides at a time. When it does bind a single strand, it straightens the DNA out from the classical B form. So the first step is not opening up the double strand target. The first step is binding the single strand with Rec-A. The actual interaction and matching up sequences occurs in the filament groove of the Rec-A itself, on the outside of that Rec-A. Large amounts of ATP are hydrolyzed, and this is another machine kind of function. You burn the ATP when you release the Rec-A. It's not required, but it really facilitates the reaction. The high affinity ATP is present, low affinity when absent. Low affinity means it now disassociates itself from the complex, from the DNA. Plecto, Greek for twisted. Initial heteroduplex may not be plecto, could be paranemic joint. 2 strands lie side by side without intertwining. I think that is a question on homework and maybe exam. I'll try to trick you up and say it's required. ATP hydrolysis is required. Not required, it just helps it out, facilitates. This is the triple strand structure that must form before you get a displacement of one of the strands. Strands are invaded, displaced, and compared with respect to strands of evading at the Holiday junction. Here you see the Rec-A. What's going on here? Paranemic. These are another fun facts that sometimes appear on exams. The paranemic, the side-by-side alignment, is not very stable, as you might expect. Can be easily destroyed by 5 degrees, 20 degrees below the melting temperature of DNA. So you can easily disrupt it just by warming it up a little bit. Here's another fun fact that's sometimes on exam. How much homologous region do you really need to get homologous recombination to work? The minimum seems to be about 50 base pairs of common sequence. Just that much, and recombination can happen. Nicking, is it required to initiate synthesis? Is not required to initiate synapsis, but for actual strand exchange, you're going to have a 3' end. Here you see strand exchange between single strand circular and double strand linear. Here's a double strand linear here, invading a single strand plasmid. This can happen in vitro. Okay, so that's the end of Rec-A. Not a whole lot left here. Let's talk about how this branch migrates and how the holiday structure forms. How much time do we have?
30 seconds.
30 seconds, some say less. Most say less. I guess less wins. We'll start here at the RUV, not rec, but RUV.
Please click back one slide.
Do what?
Go one slide back.
Okay.
I didn't get that close.
Okay.